The New World screwworm fly, often just called the screwworm, is a species of parasitic fly. It's best known for causing devastating losses to wild animals and livestock, which in turn had significant economic impacts on ranchers. Through much trial and error, two entomologists, Edward F. Knipling and Raymond C. Bushland, led pioneering research starting in the 1930s. Research that would eventually bring this parasite under control. And their work was largely inspired by research done by geneticist Hermann Muller at UT during the 1920s. What do screwworms do? Female screwworms lay eggs in open wounds on warm-blooded animals, as well as the naval cavities of their young. The eggs hatch into larvae that feed on the flesh and body fluids of the animal. The wound will become infested and seep, attracting other screwworm flies to lay eggs. The result is a complicated infection that can kill the host in a relatively short matter of time. Livestock weren't the only animals these pests infected. The screwworms also brought down wildlife like deer and squirrels, as well as domestic pets, and occasionally a human being, although very rarely. As livestock ranching expanded across the U.S. in the 1900s, so did the screwworm problem. By the 1950s, the devastation to livestock ranchers was around $200 million a year, which is about $1.8 billion today. In the late 1930s, Knipling and Bushland had become collaborators, observing and performing experiments on the screwworm flies. Knipling observed that the female mated once for her entire life. Males were different, however, and mated as much as they could. Knipling and Bushland imagined that if there were a way to make males sterile, then the female fly would possibly mate with this infertile male, and her one mating opportunity would thus produce no offspring. The researchers coined this hypothetical method as the sterile insect technique. When they proposed this idea, their colleagues considered it pretty far-fetched. Along came World War II, and Knipling and Bushland became preoccupied with researching diseases like typhus that affected the troops. However, sometime after the war, Knipling and Bushland returned to the screwworm problem and attempted chemical sterilization, but without success. Enter Nobel laureate Hermann Muller. In part 2 of our series, we looked at the genetics research Muller and colleagues did at UT from 1920 to 1932 with the fruit fly, Drosophila melanogaster. Muller reported results in 1927 which showed that mutations of genic material can be induced in Drosophila by exposing them to X-rays. After the horrors of World War II and seeing firsthand the effects of radiation, Muller was very vocal about the danger of its abuse and nuclear war. Knipling read a paper by Muller in 1950 in which Muller expressed these views, and in it, he referenced his earlier work that showed excessive amounts of radiation made fruit flies sterile. Knipling wrote to Muller and described the sterile insect technique. Muller replied that the theory was sound. Knipling and Bushland wasted no time. They found an Army hospital X-ray machine and used it on some male flies. They experimented with different radiation levels and eventually found the right formula. The efforts following this expanded in scope. After failures running tests on a small Florida island, they went to Curaçao, a small island in the Caribbean that had seen their goat populations decimated by the screwworm fly. The experiment worked and eradicated the fly from the island. The sterile insect technique then moved to the U.S. in 1955. By 1966, U.S. officials had created a, quote, barrier, unquote, against the screwworms and declared them eradicated from the U.S. The following 40 years would see this program implemented through Central America into Panama. However straightforward as this sounds, there were a lot of hiccups that would make premature this initial 1966 declaration of being fly-free. Richard H. Richardson, a UT professor, researched the eradication program in the 1970s. He was well suited for this, as his later research focused on the interactions between environment, economics, and agriculture, particularly management of rangeland and prairies. However, using the term eradicated was problematic for him. Reinvasions would happen all the time. In the 1970s, screwworms would continue to be an ongoing issue, albeit not at the scale it was in the 1950s. Still, Richardson felt the declaration of victory neglected the potential of other strains or species of screwworms moving out of Mexico and into other regions. So in 1978, Richardson and colleagues would get involved. Previously, Richardson had been studying cryptic species of Drosophila and suspected there were also cryptic screwworm species. He addressed his concerns in a book titled The Screwworm Problem: Evolution of Resistance to Biological Control. Fortunately, many of the concerns Richardson posited in this book have not materialized, but the analysis of potential problems of the eradication program were nonetheless important contributions relevant to many genetic approaches to pest eradication. Today, there is a barrier maintained in Panama through a joint commission of Panama's Agricultural Department and the USDA, known as COPEG. The modern plant, located about 20 miles east of the capital on an old sugarcane plantation, breeds 2 billion flies per year and prevents reinfestation from wildflies from South America. It's a remarkable testament to the power of cooperation between countries, a cooperation that saves American farmers alone $1.3 billion annually, and a cooperation that must look beyond politics to science.